everybody, and welcome to today's Dementia Researcher Salon Debate. I'm Adam Smith, and I'll be your host for the next hour. Thank you for joining us here in the Dementia Researcher community. I'm going to start by offering a qualification that this is our very first debate of what is going to be a 10-debate series leading us up to Christmas. And it's also the very first time that I've hosted a debate. And from chatting to our brilliant guests, Michael and Loukia, who you're welcome to turn your cameras on now, the pair of you, I gather it's their first debate as well. So this is an experiment. We'll see how it goes. We might change the format for future ones, but please bear with us then as we go through this. So today, we're going to be debating a topic that continues to generate discussion across the field, which is this house believes that animal models have outlived their usefulness in dementia research. Speaking for the notion, we have Professor Michael Coleman, a Geest Professor of Neuroscience at the University of Cambridge, whose work explores how protecting axons and synapses could prevent neurodegeneration. And speaking against the notion, we're joined by Dr. Loukia Katsouri, who is a senior research fellow at the UCL Sainsbury's Wellcome Centre, whose research focuses on molecular mechanisms driving tau propagation in Alzheimer's disease. Thank you both for being here today. Welcome back, of course. For anybody who's new to joining, Loukia and Michael have both presented in our salon before on our previous series, so do go check out those recordings. So I'll start by explaining the format. Here's how today's debate will run. Each speaker is going to start by giving a 10-minute opening statement to present their case. We'll then move on to 15-minute exchange and rebuttal, where I'll invite each to respond and challenge on the other person's points. After that, we'll have a 15-minute audience Q&A, where I'll get you can post your questions using the Q&A button, and I'll put those to our speakers. But to begin with, I'm going to start by inviting everybody watching to complete our poll so we can gauge and understand what your current views is. Now, we sent this poll out in the reminder for people to join us. And here we go. So here is a poll. There's a link in the chat. I'm going to give you 2 minutes to complete our poll. If you haven't already, don't vote twice for those that have. But we sent this link out with the invitation and the reminder, rather, about an hour ago to ask what you think now at the start of the debate. So just a reminder of the motion, which is this house believes that animal models have outlived their usefulness in dementia research. And these are the results live on the screen. So this is looking good. 27% so far undecided. So they're open to be swayed by your arguments. Only 9% agreeing with the motion. So Michael, you've got plenty of people to work with there. And we can see the numbers adjusting in real time. And there we go. I think that's probably all the votes we're initially going to get. So thank you very much. Well, a couple more coming in. But what I'll do is I'm going to leave that poll open, and we'll come back to that at the end. So to start with, let's get going. Speaking for the notion again, we have Professor Michael Coleman. Michael, the floor is yours.
Good. And I'll just start by saying thanks, Adam, for organising this. Regardless of how this plays out, I think it's a really useful educational exercise that hopefully will make all of us think a little bit deeper. The next thing I need to do is head off any accusations of hypocrisy, because as a number of people will know, my group and my entire career is based on began with animal research, and we still use animals today, especially as a source of tissue for cell cultures. And we have also used animal models of dementia in the past. And so I'm not quite going to speak fundamentally against the use of animals in research, but I will put a very strong case that we need to do so with very great degree of caution. And I'm not sure that caution is always exercised. And I think it's a very useful thought process to go through. So Alzheimer's and other dementias, of course, terrible conditions. we need to do something about them, and we as researchers, in theory, should be able to do something about them. Human genetics, in particular, through gene mutations in causing dementia, various kinds of dementia, have given us some strong leads, especially for the inherited forms, which seem to hold clues to some of the more sporadic forms. So APP mutations driving A beta and tau mutations driving aggregation and so on. They're really important clues as to what may go wrong in the brains of people with Alzheimer's, frontotemporal dementia, and other disorders. But having made that progress in human genetics, I think it's fairly clear, I think we all have to accept, that we can't go much in humans beyond observation. Of course, there's new technologies like brain imaging and so on these days, and the advance in blood-based biomarkers and so on actually are major advances. But we can't do experiments in people beyond sort of very superficial things, or if it's clinical trials, it's got to be previously tested in animals anyway. There's a legal requirement for that, among other things. So we have to have model systems. The question is which model system, and what do we get from each one, especially because today there are increasing sophistication of human based systems, human iPSC derived 2D cultures and 3D organoids in particular. So on the back of identifying, for example, APP mutations in the late 90s, early 90s, I think it was, and the tau mutations in frontotemporal dementia and other conditions, the most obvious thing for researchers to do at that time was to put them into mice. In the early days, overexpressing transgenic mice, to put that transgene in anywhere with those mutations. And lo and behold, there appeared plaques with APP mutations and tangles, or something like tangles with tau mutations. It didn't get off to a great start, however. I was in dementia research at that time, and I remember, although I couldn't, it must have been written out of history because I couldn't find it on Google this morning, the very first paper that reported that it was an obvious thing to do was to make mutant APP, put it into transgenic mice and see, could you see plaques? The very first paper said, yes, we can, and it was fraudulent. It wasn't a great start. And I think that although the correct work has been done since then, it's a note of caution about how easily we can be led to sort of believe what we want to hear, and we always need to be cautious about that. But this was a major advance. I have to say, prior to that, as I was doing my PhD, the best we had in terms of animal models was stuff like giving aluminium to rabbits or something. I mean, it's just insane now when we look back. The advance that's made, being able to put human disease genes into animals, in particular mice, has been absolutely huge. It is, however, almost always mice, and I think we should think about why that is. I think it's cost and the time, generation time, etc., play a big part in that. But we need to be aware of the caveats of only using one particular species, especially a species that doesn't develop Alzheimer's or plaques and tangles naturally. But indeed, superficially at least, these animal models have developed plaques and tangles, and this remains the only way we can study the effects of plaques and tangles in a physiological setting. However, the reason those plaques and tangles appear are not the same reasons necessarily that they appear in most sporadic cases of Alzheimer's disease, where people don't have these gene mutations. And we do need much more focus on where does that A beta and tau aggregation come from when there isn't a genetic mutation? There's nowhere near enough work on that. So sounds like I'm agreeing. And before Loukia thinks she's getting an easy ride here, I think I need to move on to some of the limitations. Now, I don't know. I will attempt to share my screen, but whether it's going to do it, I don't know. It's not going to do it. So I will just talk this through then. So there is this saying that all models are wrong, but some are useful. And I think that's very much where I stand on this. There are uses of animal models in research, but we need to start from a position of extreme skepticism and say, where is it useful? Where is it telling us something? Because the danger is we start to confuse them with the real thing. And I have heard people talk about my mice with Alzheimer's disease as though it is the real disease. And it isn't. It's one aspect of dementia in a system that has many caveats. So let's think about what those caveats are. So we know that there are some differences, for example, in protein coding, splicing, gene expression between animals and humans. Tau splicing is one of the most important ones. The difference in the 3R and 4R tau, especially in adult forms. ApoE isoforms is another crucial one, which I'm sure Loukia can say much more about. So mice don't have ApoE4, for example. Many of the people with Alzheimer's do. The differences in size of those animals, particularly axon length. I mean, we are a lab that works with axons, and that's very front of our minds. Our axons are so much longer. Our cells have to work, neurons have to work harder to support them. I'm less concerned about the differences in longevity because actually mice, I think, do age more quickly than us. I think a 2-year-old mouse is broadly similar to in its physiology to an 80-year-old human, for example. But to me, what I've talked about there is a tip of the iceberg. It's how we model disease. Mice are inbred, humans are not. Our APP and tau genes, tau in particular, have enormous genetic diversity between different people. ApoE genetic diversity, TREM2 genetic diversity, and all the other genes that have been associated with Alzheimer's disease. Not only is it the overtly pathogenic forms, there's many other genetic changes in those genes within different people. Plus, we all have different environments. We're exposed to toxins. We do nasty things like drinking too much alcohol. We get head injuries, some of us, as we go around. And crucially, we also contract viruses. And there are papers, one just last week, about the role of varicella zoster in Alzheimer's disease. Some very convincing data that it plays a role in reactivation of zoster in the brain. Can increase risk of Alzheimer's disease, and vaccination against shingles can reduce risk of Alzheimer's. This is human demographic data, and we can't just ignore it and say, well, it's not involved. That is my main concern. Most models hit one risk factor very hard. A genetic overexpression of APP or a mutant APP or one particular toxin. That's not human disease. The reasons different people develop dementia is probably different in every single person. The different combination of multiple risk factors that go on to cause that. And if we're only doing it in animal models, we're not going to get there. Quick word about human iPSC-derived systems, because that's the most obvious human-based system. What really woke me up to this actually comes from a different field, ALS, where TDP43 mutation is a trigger of missplicing of genes. And some of you may know this, the story about the stathmin-2 gene that was discovered in it's the missplicing of that gene with TDP43 mutation was discovered in human iPSC-derived neurons. And the cryptic splice site that is used there is not present in a mouse. You could work with mice for your entire career and never find that, which is probably one of the critical events in developing ALS downstream of TDP43. TDP43 is actually it may be sometimes with comorbidity, or it may sometimes be actually actively involved in Alzheimer's or other dementias. We're not modeling that in mice, and so we have to always remember this. Plus, at the end of the day, there's this whole concern about translation from things that have worked in animals, apparently not always working in humans. I would accept that the first sort of partial disease modifying treatment has been previously tested in mice and seems to be getting somewhere. But I think a lot of the reasons that these translation steps are failing is not about the difference between mice and humans. It's the difference in the way we think when we're using mice or we're working with humans. So I will leave it at that and hand over to Loukia. Very interested to hear what she has to say.
Thank you very much, Michael. That's a great start. So thank you for setting out your stall. And now speaking against the motion, we have Dr. Loukia Katsouri. Loukia, over to you.
Thank you. And Michael, this is quite challenging to continue after you, but I will try my best. So we know that, dementia is very devastating, and every 3 seconds someone in the world develops dementia. And, as you said, each case is a person different to another, it's a family, and, it's someone slowly losing their memory. And yet, after decades of research, we still have no cure. So it's easy to point fingers, and animal models are often the first to take the blame. And we hear, like you said, they're outdated, they don't translate, and they've outlived their usefulness. But that's not true. The question we should ask is not whether animal models are perfect, they aren't, but whether they're still useful. And the answer, in my opinion, is yes. They remain essential, evolving, and irreplaceable in our understanding of dementia. And dementia is not a single event in the brain, it's a storm of interacting systems, neurons, blood vessels, immune cells, and genes, and they're all influencing in ways we're still trying to map. And we cannot simply recreate this in a dish, even with a 3D organoids or in a computer model. And the animal models, especially the humanized Nokians and the transgenics and mice and zebrafish, allow us to observe how these processes play out in a living brain with a functional immune system and blood flow. And especially the thing that I am also interested in studying is behavioral changes. And in fact, much of what we now know about amyloid and tau neurodegeneration came directly from animal studies. And as you mentioned, most of these were not perfect. And I agree, we failed to reproduce our findings in the clinic, but they give us the first clues and the first hypothesis and the first therapies worth testing in humans. And without them, we would still researching in the dark. So if we talk about translation now, which is what most people say, yes, the mice are not translating and they don't predict human success. What we forget is that translation is a chain. The animal models have one link in that chain. So when a drug fails in humans, it's not always because the animal model failed. Sometimes the target was wrong, the trial design was flawed, or the biology misunderstood. And removing that link entirely would make human trials riskier, not safer. And also, let's not forget about ethics. The modern dementia, especially in the UK, the modern dementia research follows the 3 R's, which is replace, reduce, and refine. And we don't use mouse models because they're convenient. We use them because when it's necessary, and when the question cannot be answered otherwise. And today's models are more humane, they're more precise, and they're more data-rich than ever before. And we keep refining them constantly. Since I started my PhD, I've worked with a wealth of transgenic animal models, and in the last 6 years, I'm working with Nokian mouse models, and that in itself is ethical progress. And that's another point that is often overlooked. The animal research is not static. It's evolving and fast. And thanks to technologies like CRISPR, we can now create models that express the human genes and the human disease pathways. We can have a humanized mouse model that expresses the human tau under the whole locus of tau, so we get the proper splicing. We can get the humanized Nokian, we can get human complement genes. And moreover, we have technology now that allow us to watch degeneration unfold in real time, such as in vivo imaging. And one thing that is particularly close to my heart is AI-assisted behavioral tracking, which means we can monitor the mice in their home cage 24/7, and we can detect subtle cognitive changes in these animals that mimic human symptoms more accurately than ever, and way before the conventional experiment would allow us to do so. So I have to say that the animals are not relics of the past, but are more next generation tools, especially the modern animal models that we're currently using. And I think if we decide to abandon animal models now, we would throw away decades of refined knowledge. And we're now at a point that we're finally learning how to use them wisely. And I think, we should use them in a complementary fashion together with organoids and together with AI. And these are exciting, very complementary. But the organoids don't have immune systems, and they don't have blood vessels or behavior. And AI is learning from existing data. So we need to generate the data from the mice. So I will end with this, that, animal models are not the enemy of progress. They're imperfect, but are indispensable allies in the fight against dementia. And I think the future of dementia research isn't post-animal, but it's integrating. It's integrating animals, it's integrating organoids and AI, and we should work together with all of this to illuminate the complex biology of the brain. And I think that the mice haven't outlived, not the mice, the animal models, they haven't outlived their usefulness, but they're evolving. Just our science and our understanding of dementia, we must evolve with them and use them in a more caring manner and in a more sophisticated manner. And I think I will pass on to Michael again.
Thank you very much, Loukia. That's a very elegant argument. Well made. Thank you. So now we're going to move into... thank you for both for your opening statements. We're going to move into our rebuttal and exchange period, where each of our speakers is going to get an opportunity to respond to the other's introduction, and then I'll question them over a few points, and we'll move into a chance for you to ask your questions. So do start posting your questions now. But first of all, Michael, I'm going to give you a chance to respond to Loukia's arguments.
Yeah. Maybe I need to ratchet up the attack slightly, because I've been very, very balanced, I guess, or it feels to me like I've been balanced, at least in my introduction. I think one thing I'd say is, when we as humans develop dementia, we do so in a very complex environment. We've not only typically lived to middle or old age before that happens, not always. Of course, there's some early onset cases, but we lived a long life. We've done all sorts of bad things to ourselves. We've had unfortunate events happen, like head injuries and perhaps viral infections and so on. And this is simply not the environment in which we keep our mostly mice, occasionally rats, and very rarely any other species. So it really is so artificial, and consequently many of the risk factors are not reflected there. Before any of the human iPSC or organoid people start feeling smug about their research, exactly the same is true of that research. In fact, even more so in a way, because the stuff that's going on in a dish is even more artificial. There's not even in contact with the same bodily fluids that our neurons and microglia and astrocytes are. It's highly artificial. 20% oxygen instead of 4%, which there is in the middle of our brain, and all these artificial growth factors bathed in them, no gradients or anything like that. That's not the realistic for our brain. So yeah, environment is one of my biggest concerns. We're trying to model it in a very different species, and if we also change its environment, what hope is there? And the other thing that in some ways relates to that is the kind of role of information and stress. Loukia mentioned the 3 R's, which is a very important part of animal ethics, but we should be a little bit cautious about how far we go down that route, because if we prevent our animals from ever having stress, well, once again, that's not my life. That's not most people's lives. Stress is an important thing, and I actually know of a case. It's a little bit anecdotal, but a case that I know of developing Alzheimer's. The first sign came after an episode of psychological stress, and then the dementia just clicked into place immediately afterwards. And again, it's something we just don't model in animals. And perhaps just a little bit provocative. Mice can't draw a pentagon. Mice can't name 5 animals, or whatever the test is in humans, within a minute. There's all sorts of things you can't study in mice, and we need to be particularly cautious, perhaps, about the behavioural tests sometime. They also do not. It's also, nobody said so far. Maybe it's my role to say so. Mutant APP mice do not develop tangles, whereas people with HPV mutations do. So there's a lot of things we need to think about with the limitations of those models.
Thank you, Michael. I'm racking up a list of my own questions here. So, Loukia, I'm going to give you a chance to respond to Michael's.
I mean, I have to agree that the mouse background, especially if you're working with the C57 black 6, is not ideal and is not resembling the complexity that you see in humans. The environment is artificial, and we know that for a fact from studies that, for example, going back to the rabbits that you mentioned, Michael, rabbits with a dirty water in their facility developed plaques, whereas nobody could reproduce that finding, and they found out that it was just because they didn't have some iron, some mineral in the water, and it's a huge problem especially in the mouse biology. And I think one of the great things that Jackson is now pioneering with a lot of labs around the world is they're trying to use now more wild type like mice and more diverse backgrounds, and they're mixing them, and they're trying to use to introduce genetic diversity, the human isoforms of ApoE, like you mentioned. They're trying to put them back into the mouse to see more how this diversity can work in the mice. And I think there's going to be a lot of research coming out from the different haplotypes, H2 versus H1, the humanized ones coming into the mice, how one is protective and the other one is deleterious. And I'm not saying that the mice should be a panacea for everything. We do know that their inflammatory system is different, and they don't get much stress. The more stress my mice get is they walk on the elevated plasmase, which is about 50 centimeters from the thing for 10 minutes. So it's not like cycling around London trying not to get hit by a crazy driver. I think, we should understand that the mouse is a tool to answer a specific question. We have to be aware of the limitations that this model is giving us. But again, I have to go back to behavioral research. So the CANTAP test, for example, we have not developed screen tests in the mice so that they can choose objects and they can learn that and they can do it. So we're getting more sophisticated in our animal research as opposed to let's put a transgenic mouse, fill it up with plaques, and see what is happening. So I think this is my argument, that the mice can answer questions that we cannot answer in a dish because of the complexity and because of the biology. But we should always be cautious. It shouldn't be the answer to everything.
Thank you, Loukia. Great. So you've both set out your stalls and responded to each other's comments. I'm going to pick up on a few things that you've mentioned here. So, Michael, am I right in understanding your position that you agree that animal models have a particular purpose at the very earliest stages of research into a disease, for example, but there comes a time when you've learned as much as you can and you think that time has possibly come for Alzheimer's disease. If that is the case, what are the alternatives now? And are we no longer discovering new things in mice? Is it now moved into a place where we're testing treatments in mice, but we're not learning from more about the underlying disease from it?
I think unless you think very carefully about how the animal studies are designed and interpreted, I do think you reach a point where you can't find out anymore. But that doesn't mean there's no hope, and because in theory we can use our brains and and think about how we improve and take on board some of the other factors. Can you hear me okay?
Yeah, absolutely.
Yeah.
Do you think that's where new technologies take over, that you learn so much from the initial biology and then...
Potentially. Loukia mentioned, for example, humanizing mouse models. So putting in ApoE4, for example, a knock-in, or ApoE3, and I mentioned, of course, it's a related field in ALS. I mentioned the stathmin-2 cryptic splice site. So I think that there are animals now that have been humanized in that respect. So they're putting in the cryptic splice site. So the TWP43 loss does drive that cryptic splicing. But we need to be so cautious before we dive down the route of thinking this is the disease, or this is going to tell us everything we need to know. There are things you will never find there. That cryptic splicing would never have been found, of that gene, would never have been found in a mouse. So I think there are real limits on any specific question of how far it can go. We lost Loukia.
We have lost Loukia. Yeah. Hopefully she can get back in there. And I'm conscious about giving too much airtime to one speaker over the other. So, Michael, how far do you think in vitro and in silico models can really replicate the complexity of a living brain?
I am, to be perfectly honest, I'm equally cautious about that. And we also now use human iPSC-derived models. People in our building use organoids. It's always on our horizon that we might do so at some point. I'm conscious of the fact that they also, there are assumptions there and caveats there. I mean, that a 2D iPSC-derived culture makes the assumption that it's cell autonomous in whichever cell type you've got in the dish, and it isn't. And actually, even organoids make the assumption that it all happens in the brain, and it doesn't. We all know there's, the consequences of systemic inflammation, for example, impacts onset of dementia. So I think all of the model systems got some limits, but some of those limits in iPSC models are different from the limits in mouse models. So in theory, the combination is the best thing.
Thank you, Michael. Loukia, thank you very much for dealing with your technical problems. Michael seemed to place a lot of force behind the view that the mice are the same, that they're genetically engineered, that they're all fundamentally the same, and that it's a limiting factor that you can't then that the brains won't differ because they haven't been subjected to the same problems that people have. How do you I mean, you mentioned that there are humanized models now. Is there a reason why that problem exists? Can't you experiment on different mice? Can't you give them different lives beforehand?
So I think one of the problems with animal research is a lot of the mouse models were developed on a C57 black 6 background, for example, that we now know it's quite resistant to dementia. I think we're trying to now fix this by using, there's a panel of 16 strains that are interbred at axon, and they're trying to mimic this complexity of at least the genetic background. Now, the environment outside, we can't suddenly start, hitting the mice or causing stress, and I can appreciate that. We can't really replicate this in the mice. But I think even just the genetic diversity, if we can see, for example, how in different haplotypes of HLA will respond to a treatment, will give us some sort of an idea how people with that specific haplotype would respond to a drug treatment. They're not ideal, and I don't have a good answer to it. This is, I think, the strongest argument that we can't really replicate environmental and genetic diversity and all of this complexity that we see in humans, or the stress that we ourselves introduced ourselves from work, from family. I don't think the mouse has a stress about not publishing the next paper, for example. So yeah, I think this is something that we have to bear in mind when we try to anthropomorphize our findings. So you can't really call anxiety in a mouse. It's anxiety-like. It's one aspect of anxiety, but you can't say that this mouse has.
Although these do seem to be the arguments for why that model is fundamentally flawed, which could argue that continuing to use animals is a little bit like, continuing to use petrol cars. We do it because it's what we've always done, and they exist, but the alternatives are more expensive or more complicated, or there will come a point when we'll move over. Things like, I guess, recreating artificial intelligence models that could tell you everything that you could from a mouse brain. Is that not the case that you do? Are you telling us that you will continue to learn from this, that there's no alternatives at all?
There are alternatives, and I think it should be complementary. So, for example, you mentioned AI, and like I mentioned before, AI needs data. So we need to generate that data with animal models or potentially with iPSCs, and then we can fit all of it together. But if we suddenly abandon animal research, then we won't have good enough data to fit to the system. And yeah, it could be that the model is flawed, but again, you have to remember that it's only answering one aspect of the question that you want to ask. So if you think of it as a perfect Alzheimer mouse model, then you're doomed. You're never going to be able to understand it. If you think that it's an Alzheimer-like model that is answering this particular question that I have, then it's different. You can interpret it differently.
Okay. So, Loukia, could you see a future where animal models and these new tools, I mean, it sounds like you're already there, but they work side by side rather than replacing each other? Is that what you're...
I think that's what we're doing, and definitely in our lab, we're using a lot of refinement. So we're using a lot of home cage monitoring that we can do. For example, that means that we're minimizing the mouse numbers that we're using. We're trying to do, for example, after behavioral, we do histology and we can correlate. So all of this is giving us immense power to answer questions, and also it decreases the numbers that we're using because we can monitor, for example, the animals throughout their lives, their early phase, their late phase. It's quite powerful.
I understand. Michael, would you agree? I mean, are there better alternatives now than this?
To be perfectly honest, for models, on the grounds that we need models to go beyond observation, I think mice and human cells of various kinds in culture do play two key roles. Neither gives us, they both have all sorts of limits, but we should never stop looking to improve them. Of course, the development of 3D organoids in culture was one step towards improvement in the last decade or so. On the animal side, I do think there are important questions to ask about which species we use, and crucially the environment. I mean, I said it already, but I will always go on about this: the environment in which we keep these animals, and this genetic diversity. So Loukia mentioned, for example, the I can't remember what the name of the Jackson Labs panel is. The 24 strains or so F1 crosses or something like that, isn't it?
Yes.
Yeah. So there is a bit of genetic diversity there. but it's a tip of the iceberg compared to human diversity.
Yeah.
And so, if I understand it rightly,
Yeah.
the issue here is then that could become a limiting factor, because
It
actually
is. It is a limit.
We've gone as far as we can. That
Yeah.
we've learned what we can, and unless we do more, the problem is that we then reach a tipping point. That animals have,
Yeah.
that we've done as much as we can with what we have.
Yeah.
So I'm feeling
could
like there's
find-
some
we could
middle
find
ground
every-
here, which is, I think you're
Yeah.
open to arguing that there is some potential continued future use of animals, but only if we change the underlying model of the system. Or,
Yeah.
but while we ever have animals still there, is that stopping us progressing more rapidly in the other areas we could? Like, back to my car analogy, that
Yeah.
while ever we've still got petrol cars available, we're never going to fully
Yeah.
transition over to other alternatives.
So something sort of like this, you could take an APP mutant mouse and find everything there is to know about how APP mutation drives the accumulation of A beta and the formation of plaques and what that does to synapses and et cetera, and find absolutely nothing about what drives the accumulation of A beta in a sporadic patient. And there is a clear limit to how far that angle of questioning can go.
Thank you both. We're going to move on to some of our audience questions now. I think there's only Jack asked questions, so encouraging others watching to ask questions. Michael, a question we've been asked is, you mentioned fundamental differences in how species age and whether they develop Alzheimer's-like traits naturally. I think I can possibly... No, I can't. Traits and naturally, which would need to be accounted for. Now, human iPSC-derived models often represent early development stages depending on the type of cell rather than the aging brains. How do you see these systems overcoming the limitations, especially for a disease like Alzheimer's? I'm trying to find a way to pop that up onto the screen, but I couldn't. Sorry.
Yeah, sorry, I'm just trying to...
Oh, I can.
Is it in the live Q&A? It should have been there.
Here we go.
Yeah.
I can, but unfortunately it spotlights the whole thing.
I can see that, yeah. Can you just scroll down so I can see the end of it?
Sorry, apologies. I can't, because it's already
Depending
full
on
on screen on
Okay.
mine.
Yeah,
Depending
that's
on the type of cell, rather than aging, I can see that bit.
Replacing so this are you can you read the first part of the question?
Oh, sorry, I can see the whole thing on the Q&A. One moment. How do you see? Okay, how do you see? Yeah, how do I see human systems, iPSC derived, for example, overcoming the limitation about how different species age? I would say I've been very clear already. There are limitations of the human iPSC systems as well. Again, I hope I'm not being too hypocritical because we're also now working with them. But I fundamentally do not believe in the whole idea that you can age an iPSC-derived neuron or possibly even an organoid. I don't see that as aging. You're still not. It's still weeks or months, possibly even a year, as opposed to 80 years. And these are now human cells, so they don't age at the rate that mouse cells might. So I'm not sure to what extent that overcomes the age-related changes that do or do not happen in different species. I think, I mean, it's tricky because we're not using species other than mice, like especially non-human primates, is extraordinarily expensive, a lot of ethical questions. But I do feel, purely from a scientific angle, if we want to do really good science, we can't really ignore that kind of thing. There's a lot of the ethics has been mentioned already. That there's a lot of ethical questions in all this, and it's difficult to know where we each come down on that.
And I think on his third point, I think what you're both arguing is that you can't do that in either. That either in mice or in vivo models, that you can't recreate those environmental and systematic aging processes. And I suppose that's where building incredibly complex machine learning AI algorithms might be the only opportunity to do that. But you'd need, that's going to take time, and you need to give it a lot of information. Loukia, you asked, do you believe that current animal models can adequately model the role of human-specific factors like education, social interaction, complex environment exposure? And I suppose this brings me back to my point is I don't suppose anybody's suggesting that you can, but it's while ever we can't do that, then that we're just stuck at the same point. We're not progressing because we're not pursuing alternative ways to do this more rapidly, because we're just continuing to work with the status quo that we have.
Yeah. So the argument against, for example, complex environmental exposure that one could give is that we can, or social interaction. We are studying in our lab how a singly housed animal model is behaving in terms of its anxiety, stress, movement, sleep, in comparison to mice that are all together in a cage. We can make the home cages more complex and give them what we call environmental enrichment. So the mice are in a mouse paradise, and they can play and they can run up and down. And running is one of our biggest, for example, deterrent of cognitive impairment. It's not, we cannot study education for sure, but I think one of the arguments that we can model a human disease, we cannot do it perfectly, but we're doing the best we can to mimic this. And I think, again, we have to be aware of the new technologies that, are available to us, and be considerate of how we use them. And I think one of the ethical arguments about, it's not ethical to use mice for research. In my opinion, if we stop doing, for example, testing in animals, then is it okay to move it into humans? Again, we have to think a little bit about this.
Well, that opens up a very big moral and ethical question. We've kind of mostly steered away from ethics in this, on the basis that I don't think anybody here, or certainly audience, I know that there will be people who talk about the moral obligation of using animals at all in the first place. But that wasn't, obviously I wasn't planning for the debate to talk specifically to
but I think it's a counter argument. I've had people coming to me and they say, why are you using animals?
Do you think, Loukia, we continue to use animals because we're stuck, because you don't have an alternative? That, as Michael said, there are so many complexities that you can't recreate in mice, that mice brains, the biology of them doesn't play out the same as humans. You haven't got an alternative at the moment, so we just continue to do the same thing, because if you don't do it, there's no way to progress the problem that you have.
I could be a bit provocative and say that we have animal models that are developing Alzheimer's disease, and these are monkeys, the macaque monkeys, that they do sporadic form that looks like the human disease. But again, it's a matter of ethics, and it's a matter of being too expensive. And also, like Michael said, these mice are bred for research, whereas the macaque monkeys are not bred for research. They are, they're kind of like being taken. So this is a dangerous kind of terrain that I'm now treading onto.
It's time as well.
Yes.
We're all in such a hurry because we've all got, well, not all, but many have only got 3-year or grants or whatever. Yeah. And so the length of time to age a monkey is very, very long. I don't know exactly what it is. It's more than 3 years. But even a mouse, I've made the argument that actually a 2-year-old mouse is old, and probably has some similarity to us. But we don't use 2 years, because we're all in such a hurry, and it's extraordinarily expensive.
That's correct.
So we use young mice with all sorts of things messed up in them. And so we're missing aging, which is the biggest single risk factor.
Yeah, I think the best approach is to use the tools that we currently have, our PACs, our animals, answer our fundamental questions that will help us narrow down what we need to do for the next step. And I think many researchers are now aware that, we're learning a lot from iPSCs and from mice, but for some aspects we need to move on. So for example, if you want to test an aging-related drug, you need to age your mice.
So just a reminder to the audience, there is a chance for you to make your points or questions, but we're coming up toward the end now, and I am going to give, I'm going to go to Loukia first of all this time to make your closing statements. But if you'd like to make a start on that, then we'll come back to the final question. But if you'd like to make your final closing remarks, Loukia.
Yeah, so I think the research using animal models of dementia is essential, and it still has its usefulness. I think we have to be very aware of the limitations of the systems and of the alternative models that we can use to animals, and always be considerate of time and of animal suffering. We should never, ever do something to the animals that would cause suffering. So I think, if you're in dementia research, you also have to take into account aging. So you can't just use fast transgenic mouse models that recapitulate one aspect of the disease. This is not going to answer any of the questions that you have. So I think we have to use a conjunction of mouse models and AI and human data that we have now, wealth of human data coming through the biomarkers or through the studies of the NAN study in the US, for example, or the UK one. And I think we have to be, a bit open-minded. If you're stuck into no, animal model is the solution to everything, then we're never going to solve this.
Thank you, Loukia. Michael?
Yeah, I think I'd sum up that, probably everyone's spotted already. I'm not completely against, I'm not completely dismissing the use of animals in research. But I would say there's the real problem with how we think about them. In the extreme, when people talk about mice with Alzheimer's disease, and I'm borrowing again from a different field here, multiple sclerosis research, EAE as a model for multiple sclerosis. I think of all the different animal models of human neurodegenerative diseases, EAE has to be one of the worst. It isn't multiple sclerosis. It again resembles aspects of it. And I've, for my sins, some years ago I was the chair of the AWERB in my former institute, and when I was presented with a proposal to work on EAE, I used to ask, what are your thoughts about the limitations of the model? And I used to get the answer, well, it's the gold standard. For calling EAE, for goodness sake, the gold standard of a model of disease, give me a strength. We got to be so cautious and so careful about how we think about this. And there's been mention of ethics a number of times, and I think there's an important point here. And we have to, as Loukia says, we have to be cautious of animal welfare and thinking about, we shouldn't be doing things that needlessly expose animals to suffering. But if we go, I think the danger is we become so focused as a scientific community on being a bit defensive about how we use animals that we've stopped to apply critical thinking to how we do it, because there are very important questions to be asked about how we use it and how we interpret the animal research. And because we're feeling a little bit attacked from outside, not just by the kind of animal liberation kind of process, but also to some degree by the hoops we are made to jump through by the government, partly in their name, we become very defensive and pushing it, and we stop thinking about what its limitations are. And there are limitations, and we need to do that. We need to think more about that. And if we do more of that, in my view, that would move as a step that would actually improve the likelihood of translation into the clinic of what comes out of our animal research once we start to address some of those limitations.
Thank you, Michael. So, great. Well, it's time for our audience now to cast your vote. I'm going to share my screen with you. And here we go. So there's a link in the chat. It's time for you to have your final, cast your vote. Has the debate changed your view? Please vote now. We're going to give it a little bit of time to come up. You'll recall that at the start of the debate, this was where we stood. 65% agreed with the motion, 29% were undecided, and 6% agreed. So hopefully that poll is open. And there we go. Let's get our votes in now. It looks like the number of people still in the room compared to before is slightly different. So we're going to get fewer votes, I think, as people have had to disappear onto their day. I mean, it's been interesting. I think what I get a sense of here is that there's definitely a middle ground between both of you. I think I think everybody agrees that this is a complex issue, that there is, as it stands, no perfect model, that there are limitations to mouse models. There are definitely limitations to the alternatives as well. But there are alternatives out there, and that we need to clearly address the problems we have with the system at the moment to progress where we are now. So I'm just going to flick back to the previous ones just to reply you again. So previously 65% disagreed. That's now increased to 73%. But what we've done is, I think we've moved people from being undecided. Previously our undecided was 6%. We've swayed some of our undecided who seem to have been swayed more to disagree with the motion. And Michael, I'm afraid it looks like fewer people agree this time around.
I'll let you into secret. I'm one of them.
You've been persuaded as well. I think what made it for me was, I think, hearing how the use of animal models hasn't stayed static, that there is an appreciation that it needs to get better and that there is efforts to make it better, I think is a good argument. Had we been here a few years ago, maybe there would have been a different view. So we're just going to go back again. This is our start opinion poll at the start. 65% disagree with the motion, and now it's 73%. We still have a few people undecided. We've still got 9% undecided, so they're on the fence, but we're slightly of the view now that... So the motion of, if I remind myself, the House believes animal models have outlived their usefulness, has been beaten today by 73%. Disagree with the motion. Thank you very much. We're out of time now. Many thanks to Professor Michael Coleman and Dr. Loukia Katsouri for joining us today, and for all of you for taking part. It looks like opinions have shifted slightly, which just shows how engaging this discussion has been, and it's certainly given me a lot to think about. And don't forget, you can continue this discussion in the chat below. If you're watching this on catch up now, you can continue this discussion. Michael and Loukia are both members of the community, and we'll see if you tag them in posts, they'll get to see what you've written. But do join us again in the salon next week, where we are going to be asking the question. We're going to be looking at the House believes the House would redirect funding from biomedical research to social care research. I'll gauge your opinion now. Where would you both stand on that? Both as biomedical researchers, would you disagree with the House on that one?
I can go first. I think based on, stealing from what Michael said, I think if we invest in social care, we would have happier people living with dementia, and I think it would definitely improve their, let's say, phenotype, like how the disease is developed. I think, lack of social care has a really, really negative impact in patients and their families.
What about you, Michael?
So there's no question they're both underfunded and they're both incredibly important. I think what I would find really sad is if we channeled all the money or too much of the money into social care and not into research. We are essentially admitting defeat. We're saying, we're in this forever. There's nothing we can do with research, and I don't think we can go down that route.
Well, thank you very much. I hope you can both join us in the audience next week. But yeah, do join us next Wednesday at 8 p.m, where we've got Nancy Brown and Soraya Mefta, both from the University of Edinburgh, who are going to make the case for that one. Thank you very much, both of you again. And we'll see you next week in the Dementia Researcher Salon for our next debate. Take care.
Thank you.
Bye.
Bye.